Probability and statistics lie at the root of all transmission genetics and can also be applied to the study of the genetics of an entire population. Such studies help us to understand evolutionary changes in populations and inform our decisions about the conservation of endangered species. The starting point for population genetics is the Hardy-Weinberg Equation. Let us consider a simple example: one gene with two alleles. The frequency of the allele big A in the population is P, and the frequency of the second allele is Q. If there are only two alleles, then P plus Q equals 1. In order to find the probabilities of all the possible combinations of P and Q, we simply multiply P plus Q times P plus Q. This expression also equals 1 and represents all of the possible genotypes involving P and Q. If we multiply the expression, we get P squared plus 2PQ plus Q squared equals 1. This is the Hardy-Weinberg equation. To model the complex relationships in a population, we must first describe an ideal unchanging population. The Hardy-Weinberg Law tells us about the gene pool of a population. The Hardy-Weinberg Law has three parts which state that 1. In an infinitely large population in which there is random breeding, no mutation, no genetic drift, no migration, and no natural selection, 2. The frequencies of the alleles do not change over time, and 3. As long as mating is random, the genotypic frequencies will remain in the proportion P squared big A plus 2PQ big A little a plus Q squared little a, where P is the frequency of the big A allele and Q is the frequency of the little a allele. The sum of the genotypic frequencies equals 1. A population that matches these requirements is said to be in Hardy-Weinberg equilibrium. A real population, however, is subject to natural selection, so, to study such a population, we must assess the amount of natural selection occurring and factor it into the Hardy-Weinberg equation. Let us consider a real-life example of Darwin's finches in the Galapagos Islands. The Galapagos Islands are a cluster of volcanic islands just under the equator, 600 miles from the coast of South America. They can be subject to wild swings in climate, from dry periods to wet periods, to periodic intensely wet El Niño conditions. The Galapagos Islands are home to 13 species of Darwin's finches. These small birds are specialized to eat different types of foods, and these differences are reflected in the sizes and shapes of their beaks. One cluster of finches, the small, medium, and large ground finches, feed on seeds of sizes that correspond to their beaks. There is considerable diversity in the sizes and shapes of the beak in each species. In the medium ground finch, for example, the smallest beak falls into the size range of the small ground finch, and the largest falls into the range of the large ground finch. Dr. Peter Grant of Princeton University has studied a population of medium ground finches on the island of Daphne Major since the early 1970s. In 1977, the Galapagos suffered a major drought. Surprisingly, the average size of the beaks changed in the next generation as a result of the drought. At the genetic level, beak shape and size is extremely complex. Some genes will have additive effects, some will be epistatic, and a few will show simple dominance and recessiveness. Let's assume the existence of a simple gene which, when homozygous recessive, contributes to a decrease in beak size. We then express this gene according to the Hardy-Weinberg equation. P squared and 2PQ will have a larger beak, whereas Q squared will have a smaller beak. Now we factor in the fitness of each genotype. Fitness equals 1 minus the selection coefficient S. S is determined by direct observation in the field. We now rewrite the Hardy-Weinberg equation. If there is no selection, S equals 0 and fitness equals 1. The frequency of the genotype is determined solely by the frequency of the alleles. If there is selection, the selected genotype will be reduced according to S. During a very wet season, or an El Niño, plants are abundant and there are many small seeds, which are easy to crack. Therefore, birds with smaller beaks are not at a disadvantage, and there is no selection for this characteristic. So, in the Hardy-Weinberg equation, fitness equals 1 for all genotypes. In the year prior to the drought, Grant calculated the natural selection coefficient against smaller beaks to be 0.08. During the drought year, the population of medium ground finches dropped from 1,200 to 180. Grant calculated that the selection coefficient had risen to 0.3. As expected from the Hardy-Weinberg equation, most of the birds that died had smaller beaks. That is, all of the small, easy-to-crack seeds were eaten first, leaving the larger, harder seeds. Only those birds with beaks deep enough to crack them survived. When the rains returned and breeding resumed, the average depth of the beaks in the next generation had increased by 4 to 5%. This is not a complete picture of natural selection in Darwin's finches. For example, in other years, the beak sizes became smaller, and in young finches, selection favors small size because even the larger bills of young birds are not strong enough to crack the hard seeds that they will feed on later. All such factors are additional departures from a non-changing Mendelian population in Hardy-Weinberg equilibrium. To the extent that they can be measured, they can be factored into the Hardy-Weinberg equation to create a realistic mathematical model of an evolving population.